Hello and welcome to this webinar titled Use of Computational Modeling in Specification Setting and Establishing Control Strategy, hosted by Biopharma Asia magazine and presented by Thomas O'Connor, scientist at FDA, followed by Pharmaceutical Quality, Data Quality and Model Quality, presented by Sean Bermingham, Head of Formulated Products at PSE Limited. My name is Stephen Edwards and I will be your moderator. Before we begin, I would just like to inform our viewers that there will be a live Q&A session after both presentations near the end of the webinar. Audience members can send their questions in at any time during the webinar via the Questions tab located directly below your webinar screen, and we will go through these at the end of the presentations. Now please allow me to introduce our first presenter. Dr. O'Connor is a senior chemical engineer in Division of Product Quality and Research in the OPQ's Office of Testing and Research, and is a member of CEDA's Emerging Technology team. His responsibilities include coordinating and directing regulatory science projects to support the implementation of emerging technologies. His team's independent research has centered on advancing the utilization models in pharmaceutical development and manufacturing. He originally joined the FDA as chemistry reviewer and has served as a primary and secondary reviewer for NDAs and ANDAs. Tom is a co-author of several papers and book chapters on continuous manufacturing and emerging technology. He has consulted on several regulatory applications utilizing continuous manufacturing and is one of the government liaisons to the U.S. Pharmacopoeia Expert Committee on Continuous Manufacturing. I'll now be handing over to our first presenter. Welcome, Dr. O'Connor.
Thank you, and I'm very happy to be here today to kind of give a perspective on validation of computational models for establishing control strategies. So to start with, just a quick disclaimer: this presentation reflects my views and doesn't represent FDA views or policies. So just to give over, I'll start with this background and then go into our current regulatory frameworks for looking at computational models as part of controlling product quality, and then I'll end with some case studies. But to start off, this figure kind of represents what we view as our current sources of evidence to make decision making today, and that comes from humans, laboratory, and animal models. But as you see, as we move into the future, we see a greater role for in silico sources of evidence. Now, this slide is really put together for clinical trials, but this is impacting across the different disciplines across the FDA. Recognizing that, FDA has formed a modeling and simulation working group a couple of years ago, and the FDA encounters many different kinds of models that impact its decision making, and the working group is situated, it breaks out based on the type of modeling. So there's chemical, there's mechanistic, statistical, physics-based, there's a wide range. And the working group objective is really to raise the awareness about modeling and simulation and how it's used to advance public health and foster enhanced communication among the different stakeholders. And the working group we have about over 200 members in the FDA. But today, what I'm really going to talk about, that's across the FDA, but what about pharmaceutical quality? In quality by design framework, the models can be used at every stage of the development and manufacturing. And we have examples of predictive models that have appeared in regulatory submissions and that have been used to control product quality, such as dissolution models for release, multivariate statistical models that are used for estimating the residual solvent in a drying and API process, and of course there are telemetric models associated with PET methods that can be used for real-time release. And we've seen models in all these different buckets, from product and process design, risk assessment, design space identification, in-process controls, real-time release, and so forth. Just a quick roll on terminology and where we are and where we, I think when we talk about the evolution that's happening currently, these terms are coming to play. So most of our, in product quality, when we're talking control strategy, most of the models we experience are in this empirical. They're models derived from data, experiments, where we're kind of recognizing relationships based on the experiments you're running. A lot of the models, maybe in the process design or in product design, you can have more mechanistic elements, and there you're talking like more like first principles, and you're understanding the relationships between the variables, and that's how you're deriving the models. But we see elements of these kind of coming together. When we talk about the models we're seeing for control strategies, implemented as part of control strategies today, we're mostly looking at these hybrids, where they have elements of empirical, but there's elements of a kernel of mechanistic understanding that sets the relationship between the variables. There's many benefits and challenges when implementing models, and this is really kind of our internal perspective about, well, as a regulatory agency, we also see models as potential benefit for process evaluation and quality assessment. So can we use models in how we do our work, and not only, sponsors. And that's, it's very helpful to establish input and output relationships. I think they can be used throughout the product life cycle, such as risk assessment for change prior to implementation. But there are a lot of challenges, and one challenge for us is the access to data that's representative of the products and processes we're trying to model. But I think a common challenge that faces both sponsors and agencies is how to establish the credibility of that model. So that's where verification and validation come into play. And if you're talking about models that are going to be used as part of control strategy, lifecycle maintenance enters the equation. So as I mentioned before, development and assessment of process models by OPQ is not unprecedented, but the frequency and type of models and applications are evolving. So we're moving from where we would traditionally just see models derived from design of experiments. And when we had PAT, we had some geometric models. Now we're seeing a much more wide variety of models that are being implemented as part of the control strategy. There's still a number of applications, but this is growing and evolving. And we feel this is driven in part by the adoption of advanced manufacturing, such as continuous manufacturing and other advanced manufacturing processes. These processes are generally inherently data-rich. They're connected to plant-wide control systems, which is collecting information, because they are, new models are being applied to them, and we see, for all these reasons, it's kind of raising the profile or the use of modeling as part of the tool to implement these advanced manufacturing systems. So if we look at the current regulatory framework, we do have some regulatory documents that talk about models, and one of the primary ones is the ICH Points to Consider document, which talks about models, and it categorizes models based on impact to product quality, with high, medium, and low impact. Your high impact models are typically reserved for your sole indicator of product quality, so this would maybe a real-time release sort of model. Your low impact model would be something that maybe falls in the realm of a process design, as helping you make a decision at that point in the process, far away from actually impacting product release and certifying product quality. It provides limited amount of information, very general, about actually how do you validate models in order for these different impact levels, although it does talk a little bit about how you would document them in the regulatory submission. Now, one type of model we have gained a lot of experience with are models associated with process analytical methods, particularly NIR. And in the NIR draft guidance, there's a lot of actually more specific information about how to validate those specific models. And we talk a lot about the information we'd like to see on that external validation set, and what information needs to be supplied as part of that, and how it's validated. Because this is part of the analytical method, some of the principles of validation analytical methods are also employed. And we also talk again about this idea of lifecycle maintenance, because these are models that are typically employed as part of the control strategy when they're submitting. And while this guidance is submitted, written specifically for an AR, it's fairly general for most spectroscopic PAT technologies. But these are just, I'd say, very focused. It doesn't really address the kind of new kind of models that we're seeing as part of the control strategy. So we've been looking to other areas to gain some experience about other best practices that we could adopt when we're trying to address the verification and validation of these hybrid models or these other type of models. And this is an area where we've tried to leverage some of the learnings across the FDA. And our Center for Devices and Radiological Health has been working in this area for a while. And so this has come up a few years ago. They came up with these 10 not so simple rules, and I think as any modeling practitioner, you look at these rules, they seem kind of self-evident that this is what you would try to do. They're called not so simple because I think when the devil's in the kind of the details, and when you're actually talking about a specific case and what do you need to do at each of those levels, it becomes more tricky about deriving consensus about what needs to be done. So that helped to form part of the motivation where they said, okay, can we be more specific and provide more guidance? And that got a charter for a standard. That's the ASME Verification Validation 40 standard, which was published in January this year. But this is kind of some of the motivating factors. Here, in a regulated industry, where these models could support decision making, what levels do we need for establishing the credibility of that model? And I think there's things we can take from this framework that I think are applicable even in the pharmaceutical context. So one of them is the idea of risk. And so the kind of crux of the framework is really a risk-informed framework. So this means kind of defining, so you start with defining your question that you're asking, developing a model to address, and putting the role of that model in the context of how you're going to use it with all the other evidence you're going to use to make that decision. So that helps you kind of frame what's the risk of the model making an incorrect decision or kind of leading you to make an incorrect decision. And that's where you get to kind of the next step in that process is once you define a clear context of use, is really kind of breaking it down and then deciding the risk. And the part here, it's different a little bit from the ICH context where we had impact, which was kind of like a risk, is they break the risk down to two factors. So this is model influence, which you can kind of think as associated with the impact. So if the model is highly influential in decision, it could be the sole driver for helping you make that decision. And if you have one crux, if it's just one piece of many that you're using to make the decision, the influence will be less. But there's also another access, that's decision consequences. So that's consequences if the decision is not correct. And just to kind of tease out how this would be maybe a little bit different than in the impact case, you can have, say you have a release model for content uniformity, and you have one drug product that has a high drug load, free flowing, very little segregation risk, but because that model is used as defining indicator of quality, that would be a high impact model. And you can also have that same type of model, but in case where you have a low drug load, narrow therapeutic index, other risk factors, which make that prediction much more sensitive to impacting patients, right? And again, that would make be a high impact model. But when you look at decision consequences, I think there's more risk in those different types of products. So it just gives you two axes to look at the risk of the decision. But the next step, another, I think, nice thing the standard does is it breaks down these credibility factors. So it breaks down specific activities. So as verification, so this is kind of verifying that you kind of correctly implement the model, and then validation, making sure the model kind of correctly captures the situation you're trying to model, and it breaks it down to a number of different factors from the model, from the comparator, how these things overlap, and you can kind of use this to help you build a plan. And not that you would do each one of these activities in each one of these categories, or the level of the work you do in each one of these categories would depend. It goes back to the risk assessment and the context of use. So it just helps you kind of build a plan. So you're building a plan that's based on the model risk. And then in each of those credibility factors, there's gradations. And gradations, it's just a way to kind of describe a progressive level of scrutiny that you're applying to the model. So the standard provides some guidance on gradations, but they're pretty general. But where they come through a little bit more is in the examples they provide in standards. All these examples are related to medical devices. And I think there's an opportunity moving forward for if there are cases that can be developed in the pharmaceutical context. This is really one of the areas I think that there could be some value kind of providing more context. So here's this one case this is at the level of rigor for output comparison. You can start off with visual comparison. You kind of plot the data together. It looks like it's good agreement. Have you kind of the lowest level, and then you can be more quantitative, and then you can kind of be more stringent about what level of agreement you're accepting, and then how you're dealing with uncertainty in both the measurement and the modeling, and how rigorous you're kind of treating that, and you can kind of go down to different levels about how much work you're putting in. And that would just, again, depend on the model risk. So I think when we've looked at the standard, I don't think it's not directly applicable to the hybrid cases that we're seeing, because the standard was really geared towards these physics-based models. It's not directly applicable, but I think there's two things that I think are really useful. So there's the context of risk-based validation, making sure that it's fit for purpose, and then kind of building these plans, kind of breaking out these activities into different categories. And then I think an area where we could develop further is kind of working through these gradations and providing more examples of what that would mean. Especially, you can have two models that basically are similar, or the same model, but used for different contexts of use, and you might do different validation activities depending on the context used for the same exact model. To make it a little bit more concrete, I'd like to just wrap up with my presentation by just talking about two case studies. So the first case study is going to be design space for continuous drug substance process. This case study is adopted from an actual application. So in the continuous drug substance process, where you can have many reactions in flow kind of integrated together, this can be challenging to study by strictly the DE approach, because basically all the reactants and the solvent is traveling from the first reaction, is traveling right back into the second reaction. You're adding in more reactants to carry out the next step of the reaction. And the number of species in this system just kind of keeps increasing. And then that obviously can lead to potential side reactions. There's some dilution effects. And there's a lot of parameters to consider, both the temperatures and pressures of all the different reactors, the flow rates of all the different materials. The upstream process reaction can then impact the downstream reaction. So in this case, the sponsor took a modeling approach to really look at the design space for this reaction. And again, using a modeling approach does not mean they didn't do any experiments. They actually did targeted experiments instead of a DU approach around the whole process. So what they focused on was measuring the reaction kinetics of both the major reactions and some of the minor reaction pathways. So this is how the impurity formation. They were looking at they did some calorimetry experiments. So the model does include a heat balance and heat reactions. The system is assumed to be well mixed and flow flow behavior. So it was not kind of detailed CFD or anything like that kind of. So when I'm looking at the context of use in this case, if we put it in the framework of the standard we just talked about, if they wanted to predict the levels of impurities at the end of the synthetic process and form a design space around that, and then they were going to look experimentally confirm kind of the worst case scenarios, a set of worst case scenarios that were identified by the model. And that's how they were going to validate the modeling approach. And then what I showed actually on the previous screen, this is not, this is actually a case for ibuprofen that we've taken from literature and that we've worked on. So this is not strictly the case study, but I think it gives you kind of the idea of what a typical continuous flow process might look like. And actually some of this data is actually taken from our ibuprofen case study, not the sponsor's case study, but again, similar, showing basically how temperature disturbances can impact impurity formation or running at different temperatures. That's one of the elements they looked at in the design space. So they, as part of their validation exercises, they looked at the governing equations. So they challenged the kinetic pathway model they put together. They tried different formulations there. They looked at the sensitivity analysis of some of the key process parameters, the kinetic rate constants. In the comparator, they looked at not just one worst case, they looked at 16. So that's, and then for the validated output comparison, basically they wanted to look at both the model and the experiments were achieved. That even the worst case scenario, they were still well below the threshold that would be for these impurities that would be cleared across them. And the model DUE consisted of 537 runs, so much more than they ended up experimentally testing it in the end, just to verify those worst case scenarios where they still found that they were well below the impurity threshold level. So that was, again, a good case of kind of using this template just to structure the activities. I mean, this is not how they talked about validation, but when we were looking at it, it helped us structure how we were setting the model and kind of talking about everything they did and trying to put it in buckets. So we had a complete picture of how the credibility of that model was established. So the second case, again, a continuous manufacturing process, but this time we're going to look at the drug product process. And here is, again, an example of a continuous direct compression process where you have material loss in weight feeders feeding both the API and all the excipients. You can have sometimes just a single blender, everything kind of flows in that blender. In this case, we had 2 blenders. The second blender doing the lubrication blending, and then going to the tablet press. So one of the key things as part of developing this process, understanding this process, is understanding the process dynamics. Typically, we've seen this done by kind of measuring the residence time distribution. So the probability distribution describes the time the other master fluid element remains in that process. And it gives you kind of a macro mixing scale model of the process. And using that understanding, you can then fit that understanding to a series of well-known model classes, and then use that kind of to predict basically macro mixing. So you can take disturbances or input feed rates and then try to trace those materials, but even if you take a step further, actually predict what the concentration would be, taking in the real-time measurements of the feed rates. What would that blend look like at the exit blender 1 or at the exit blender 2? And what we've seen this used for a lot in the control strategy, again, is designing these control limits. So typically, because you have dampening in the process, your feeder rates, or your feed limits, don't need to be at, say, 90 to 110% in order to get 90 and 110% of the output of the process. There's some dampening, mixing happening in there. So companies sponsors tend to propose wider limits there, because even with those wider limits, they're still well within spec by the end of the process. And in these models have been used to kind of justify help set those limits. And so in this case, the context of use was to actually predict the formulation components in the blend, not just the API, actually also the excipients in this case, and in case there was an excursion that led to excipient being below a threshold needed. And the primary control strategy, there is still an NIR being measured, so this is not the model's not being solely used in real time to kind of measure the concentration. It's going to be used in conjunction with this NIR method. But the NIR is focused on the API, where the modeling is focused on all the factors. Again, in this case, they looked at, from a governing equation perspective, they looked at the sensitivity of the model form. And then they looked at kind of the sensitivity in some of the model parameters. If you're like in tank and series models, this might be like the mean residence time and number of tanks, things like that. And then comparator, they actually looked at a number of different process conditions and material, particularly API properties, and found that single models actually able to capture the dynamics across this wide range of process conditions and material properties. The output comparison, is kind of said here. And the applicability, they actually tested over a wider range than they were proposing in their design space. So again, this is just a framework in which we put the information needs right in to kind of just give an overall assessment at the level, and it was, well commensurate with the risk for this model. So I just wanted to share just some concluding thoughts. So our regulatory experience, as I know, as process modeling is evolving, we're moving, just having more diverse experience, I'd say, over the last couple of years than maybe traditionally, where we would typically only see models derived from design experiments for a process model. We continue to see like chemometric models and associated PET methods, but now we're kind of seeing a lot of these, like flow sheet models, which I think Sean will talk some more about in the next talk, and other kinds of these hybrid models, really to inform setting control strategy limits and helping inform that decision making. Again, advanced manufacturing, emerging technologies are kind of a driving force. I think you see that some of the examples I highlighted case studies. This doesn't only apply to continuous manufacturing, but we do see it very commonly associated with continuous manufacturing. And verification and validation of activities for models, I think this should be fit for purpose, and I think conversation will continue to have with sponsors as we propose these models, and as a community, I think we can continue to strive to kind of get more consensus about these. So the V&V 40 standard, I think, is useful, along with our current regulatory guidance. I think there are some differences in the pharmaceutical context, but I do think it has been helpful for us as a starting point. But it needs to be adapted a little bit to kind of cover these hybrid models. And I think the gradations for specific case studies and developing those case studies where we actually get into a level of detail of, what are those gradations, I think will be very useful for the community at large and FDA in turn. So with that, I would like to conclude my talk. These are some of my colleagues, Nima and Michael, who did some of the modeling work, and then my leadership, and Tina Morrison is certainly the leaders in the FDA on the adoption of modeling for decision making. So with that, I'd like to conclude my talk, and thank you for your attention.
Thank you, Dr. O'Connor. Now, before I introduce our next presenter, Dr. Bermingham, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. Please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we will go through these at the end of the presentations. Now, please allow me to introduce our second speaker. Sean joined PSE in the year 2000 and has been head of PSE's Formulated Products Business Unit since 2010. Prior to this role, he was PSE's Global Head of Consulting. Throughout his career at PSE, he has acted as a principal consultant in over 30 crystallizations and solids processing projects. In his current role, he is responsible for strategic business development, software product development, and services delivery for the pharma, food, consumer good, specialty, and agrochemical industries. Sean is a leading figure in the development and adoption of mechanistic model-based tools for increasing R&D efficiency and managing risk in areas ranging from clinical trials to tech transfer and operations. He is the driver behind QBD 2.0, which focuses on improving the efficiency of QBD by reintroducing the sound science element of the original QBD definition. He is one of the founders of the Systems-Based Pharmaceutics Alliance with Eli Lilly, GSK, Pfizer, and Roche. Sean is the lead for the £20.4 million Adopt Digital Design project with partners including Astrazeneca, Bristol-Myers Squibb, GlaxoSmithKline, and Pfizer. Sean is a chemical engineer with a master's in chemical engineering and a PhD in crystallization from Delft University of Technology and an executive MBA from Imperial College in London. I will now be handing over to our second presenter. Welcome, Dr. Bermingham.
Thank you very much, Stephen. Okay, so following on from Tom's talk, I'm going to talk about the link between pharmaceutical quality and model quality, and also the role of data quality, seeing as that's used to calibrate models. Okay. In terms of the outline, I'll give you a quick introduction in terms of who is PSE, what is our vision for using mechanistic models in life sciences, and our modeling environment to realize that vision. Then I'll talk a bit about pharmaceutical quality, QBD, the sort of more traditional QBD based on multivariate data analysis and how that compares with QBD using mechanistic models. And a virtual DOE framework that we have developed to sort of automate that QBD process. I'll show you a few applications from drug substance and drug product manufacture that make use of that framework. Then I'll move on to the importance of model quality, data quality, and also how we can actually assess the quality of a model, again, given the context of use principle that Tom mentioned, and how we can do that in a more quantitative manner. And then I'll also wrap up with some concluding remarks. So very quickly, PSE started a few years before I joined in '97 as a spin-out from Imperial College in London. We're a company that focuses on a wide range of industries, which you can see on the right-hand side, and started focusing on the pharmaceutical industry in about 2009, initially focusing on the development of unit operation models for drug substance and drug product manufacture, and a few years later also developing product performance models in the area of oral absorption and stability. We're a company of about 160 people and headquartered in London, and major offices in New Jersey and Korea. Our mission statement is to define, develop, and drive the adoption of next-generation modeling technology, methodologies, and workflows. What you'll see in this talk will be there's been a focus on putting more mechanistic understanding, more physics, more chemistry into the models. But actually what we'll focus on more is how to use those models, how to calibrate them, and how to use them to define design spaces and also determine how much confidence you can and should have in the model. In terms of our vision for the life sciences industries, our vision is to enable drug products and the manufacturing processes to be optimally designed and operated with fewer resources, so fewer experiments, less manpower, reduced risk in tech transfer, in operations, and for better end-use performance. And we aim to achieve that vision through the use of mathematical models based on mechanistic understanding, and in some cases also data analytics if mechanistic understanding is missing. So the concept of hybrid models that you also heard in Tom's talk is also something we work with. And in literature, quite a few examples now of people using mathematical models effectively to help design robust processes and products, often is done by academics or leading scientists, engineers within the pharmaceutical organizations. Making that more widely accessible requires tools that allow you to rapidly configure, calibrate, and deploy those models. So it's not just about developing models. A lot of this is about workflows. As I mentioned before, we develop models for manufacturing processes, which result in the drug products, and we also develop models for the performance of those products in terms of oral absorption or in vitro dissolution, precipitation, and stability models. Our vision is based on being able to link those models so that we can actually look at the combined effect of not only patient physiology, but also manufacturing decisions and disturbances on the final product performance in vivo, as well as understand how a certain formulation and combination of manufacturing conditions will result in flowability or not, good content uniformity or not. And by having a holistic approach to both the drug product design and the manufacturing process design to ensure that we reduce the iterations between somebody designs a very good formulation, but it's very, very difficult to manufacture. And sometimes there's no time to reformulate, so you end up having to launch something that has poor manufacturability. Of course, there's also the effects of or linkage to supply chain, which is something we are not looking at as closely as the linkages in this manufacturing to product to patient. Of course, we want to understand, if we're designing if we want to design a robust manufacturing process, we need to understand what is the likely variability and quality of different excipients, so that we don't only design the process for the excipient quality of today, but also the likely excipient qualities the manufacturing process will see in the future. And on the other end, sort of more of the scale, we also want to make sure that we have manufacturing processes that can respond to different demands for, changes in demand for certain drug products. The SPP Alliance, as Stephen mentioned in the introduction, is the driver, actually, or who helped formulate this vision for the holistic approach to designing the drug products and the manufacturing process. It's a vision that was defined back in 2012. More recently, I came across this diagram, which comes from a Siemens website, which nicely shows also the linkages between digital twins of manufacturing processes, digital twin of product performance. So in terms of realizing that vision, there's a number of elements. One is you need to have mechanistic models. And so within the Alliance and also within the ADOPT projects that was mentioned earlier, a lot of models have been developed, both on the drug substance manufacturing side, drug product manufacturing, product performance. This is very much focused on small molecule and oral dosage forms. It is being extended to large molecule applications as well, but I'm going to talk about small molecule today. So one thing is to get models that have appropriate physics, chemistry in there. The other is to have tools to calibrate the models, not only to get fits of your model predictions and the experimental data, but also to be able to look at the parameter and the parameter uncertainty and correlations, because that's ultimately the measure of the model uncertainty, and that combined with the context of use will tell us whether we can trust the model enough or not. In terms of this framework, so the mechanistic unit operation models, typically these models are calibrated on a per unit operation basis, or if possible on a per phenomenon mechanism basis. So things like solubility and isotherms are of course calibrated in isolation from the actual unit operation model. Once you have the calibrated models, you can use them to construct integrated system or flowsheet models. This is a screenshot of a model that runs, and there are one or two applications in industry where people have done this. But to set realistic expectations, what people do today is typically they'll look at individual unit operations or as you'll see in some of the case studies, they'll link a number of unit operations within drug product manufacture or drug substance manufacture. So depending on A, the need of the problem that you're trying to address, and B, the quality of models and data, you may look at larger systems or smaller systems. And you have the flexibility to do so. And then these calibrated unit operation flowsheet models can be used for optimal design and operation, sensitivity analysis to identify and mitigate robustness issues, which of course relates to sort of our design space considerations. And you can also take these models into operations for things like soft sensing or model predictive control. So pharmaceutical quality. So QBD was also mentioned earlier. I put QBD 1.0 and 2.0 in quotes because I don't think they are widely accepted definitions, but we find it useful to try and explain the difference between QBD that most people know, which is based on factorial DOEs and then using the output of the experimental program to build multivariate data analysis or statistical models, versus QBD 2.0, which is based on mechanistic models. So I'll try and explain the difference between the two briefly. So in what is considered more traditional QBD, the models that they use are typically statistical models, and the data is used to build the model. The aim of the program is to determine the combined effect of critical process parameters on quality attributes, critical quality attributes, and the approach is to identify robust formulation or operations or control strategies is to perform a physical DOE in the lab or on your pilot plant or even commercial with respect to 3 or 4 factors that are deemed to be the critical formulation and process parameters. Often those parameters, those critical parameters have been determined through maybe some qualitative analysis, fishbone diagrams, and so on, which is based a lot on heuristics and people's personal understanding of the process. Limitations of the approach, it can be very resource intensive, especially as the number of factors go up. It may increase exponentially. The limited ability of transferring knowledge to other equipment or skills, whether it's or from batch to continuous operation, and so on. And then in QBD 2.0, the models that we use are mechanistic, typically, and the aim of the program is not to determine the combined effects of the critical process parameters on quality attributes, but it's to estimate the parameters that describe physical, chemical phenomena of importance to that model. And then we will investigate critical dimensions and areas identified by the virtual DOE. So the virtual DOE is using the model, not your experimental setup, to understand the combined effect of many factors. You'll see examples with, I think, about 8 factors. In practice, we have now seen this go up to 15, 16 factors, and depending on the increases in computational capacity, it can go up a lot more. And because we can look at many more factors, we can actually use the model to identify true critical parameters based on sensitivity analysis. And then you could still decide that you want to do either a factorial DOE in the lab using the top 3, top 4, top 2 critical parameters, or you could actually say, well, I'm not going to do a factorial DOE, but I'm going to look at critical bounds of the design space that were identified through the virtual DOE. Now, this approach also has a number of challenges. First of all, they are mechanistic models, so you do need to have people who understand the process well enough to choose the appropriate mechanisms. In some case, there may be a few different postulated models for a mechanism, so you may have to do some model discrimination. Again, mechanistic models is something that is not as widely known and is applied as data-driven approaches, so there is quite a bit of training and also culture change required, also in terms of the kind of experiments that people may do. And then, from a regulatory perspective, I think both QBD 1 and 2 require good communication of what has been done to make sure that the information can be processed correctly. So you can see virtual DOE is a key sort of term here, and I'm just going to explain how virtual DOE works on the next slide. So assuming we have a calibrated model of our manufacturing process, it could be a single crystallization step, it could be a whole CDC, continuous direct compression line, and so on. If we have that model, we're going to have different factors. We have factors that relate to the process parameters or formulation parameters or a combination of the two. These are the things we choose. We need to choose the best combination of these parameters that gives us the right in vivo performance as well as good manufacturability. And when we talk about robustness, we're looking at the combination of parameters that gives us the right nominal performance, but also has the lowest sensitivity to variability in raw materials, patient-to-patient variability, any sort of process disturbance. What we used to do up to a few years ago, before I think we had automated virtual DOEs, was to run a number of different simulations. Every simulation so it would be a combination of some of these inputs, the environmental inputs, some of the decision parameters, and we'd look at the outputs, Cmax or D50, and so on. And of course, in our minds, try to create a mental map of what the combined effect is of all these changes. What we are now able to do is to look at something we call global system behavior, where we're not looking at a few points in this environmental input space, multidimensional input space, and we're not looking at a few points in the multidimensional input space relating to our decisions, our formulation and process parameters. We are now going to look at distributions. Now, they could be uniform distributions, which is what we typically will use for our decision parameters, and they can be normal, log normal, correlated distributions for our environmental inputs. And by running lots of simulations, thousands, tens of thousands, hundreds of thousands of simulations, we can look at many combinations of all those inputs, and we will get a predicted output of the predicted distribution of the output of the model in terms of again, Cmax, D50, flowability, and so on. This is what we call uncertainty analysis. If you look at this plot and you think that, for instance, the Cmax is all over the place, it's sometimes much too low, so it may not be, it'll be efficacy concerns, or too high, there'll be toxicity concerns. If that distribution is too wide, well, that's good to know, but this plot doesn't help you determine what to do to mitigate that risk. That's where the next part comes in, which is the sensitivity analysis, where we actually look at the sensitivity indices with respect to all the inputs, both decisions and the environmental inputs, and that's where we can determine, if we want to reduce that variability, is it mainly the patient-to-patient variability that we need to try and reduce by maybe having different formulations for different populations. Okay, so obviously high performance computing is a big enabler, although many people run, some of the examples you'll see were run on laptops or desktops with 8 cores, but as people want to look at larger systems with more factors, high performance computing is, as I said, a key enabler and something that is supported. The other important thing to mention here is the importance of big data analytics. Although big data analytics may not be the most appropriate tool to understand what happens within the unit operation, we do believe it's a very powerful approach for understanding what some of those inputs should be, in particular the things like the raw material variability or patient-to-patient variability. So collecting data to put in appropriate widths of distributions is of course key. If we make this too narrow, we can fool ourselves into thinking it's very robust. If we go with a very, very broad distribution of raw material variability for raw material properties, we may think that the process isn't robust at all, and we're trying to design it for situations that may never appear. Okay, so let me give you a few. I'm going to go through this fairly quickly. There's 4 case studies on that use this virtual DOE framework. The first one is an application that was presented last year. It's the whole presentation, and I think most of the following presentations are available through the PSE website that have been cleared for public distribution. This is an example of the approach being applied to drug substance manufacture and to batch. And for me, the interesting thing about this example is that it moves away from what I think is a traditional approach of batch, where every batch step seems to be investigated very much in isolation. This flow sheet here, it may look like a continuous process, but it's a series of batch steps. And at the end of the batch, this first batch will move the material to the second batch, and when the liquid-liquid extraction is finished, it will move to the crystallizer. So this means that we can actually look at how disturbances will affect, what happens in this, not only in this reactor, but also means that the liquid-liquid extractor is going to have variability in its starting point, and the crystallizer may have variability in its starting point. The first thing that was done was to calibrate the model. That's the first thing you should always do, of course. So here's some example of the kind of experiments that were done, measurements. You can see that it mentions reaction kinetic scheme 4. So there were chemists had formulated different mechanisms. The model was used to identify which one is the best to represent the various experiments. And with the calibrated model, they then did these virtual DOEs, or here you can see results of the uncertainty analysis. And this showed that, in terms of pharmaceutical quality, in terms of purity of the product, that limiting the reaction time was favorable from a purity perspective. The next case study is also drug substance manufacture, but continuous, from the same presentation. So this is also a flow sheet, but now there is continuous, or in certain cases periodic flow between the different units. Again, there was calibration. Here we're looking at stage 2B of this process, which was the middle bar part. And once the model had been calibrated, the model was used to investigate different temperatures, low, high temperature, low and high flow rate. And of course, they found out that they were able to look at the variability in predictions. And here, unlike the previous example, they also looked at the actual sensitivity analysis and found that which of the factors were the top 2, which is here the flow rate and something related to composition. The next example is an older one, a few years old now, Astrazeneca, and this looks at roller compaction. Looks at the roller compaction step, so formation of the ribbon, milling the ribbon into the dry granules. There's a lubrication aspect, and then looking at the tableting, the compaction step, to look at the predicted tensile strength, porosity, etc. So this combined model, actually the calibration, again, if you get the whole case study from the PSE website, you'll see that the calibration was done individually on the ribbon formation, on the milling, the lubrication, and on the compaction. That gives you a better estimate, less correlation. It also requires less material than, of course, trying to fit it to experiments on the integrated system. With the calibrated model, they set up a virtual DOE, or experiment design, and you can see there's a number of factors here, a few factors from the roller compaction stage. Actually, 2 of these factors are decision parameters, and one is is a disturbance, a variability, side seal leakage. And then the other three factors on these 3 unit operations are all also process parameters. Interest in looking at a number of responses: tensile strength, porosity, some particle size, distribution of granules, flow function, coefficient, porosity, and so on. So this again was set up, so automated calculation. 3,000 simulations were done on a laptop or a desktop computer. And this calculation was done for different grades of the lubricant. So the top left diagram, the 9 diagrams top left, is for one grade of excipients, and then the top right is for another grade of excipients. And if you overlay the two, you get the diagram with the figure with 9 diagrams at the bottom in the center. And the areas that are still light green are areas where you satisfy your 2 quality attributes, regardless of the quality of lubricant. So this allows you to, when you convey this process here to commercial manufacture, give people much more confidence in their ability to deal with different grades of raw materials. The final example is one that was presented quite a few years ago at ASCHE, and some of you may have seen updates to this. But this is an example of using the mechanistic models to. Not sure why that happened. Looking at mechanistic models of blenders, feed frame, surge hopper, and taking measured time series of feeders to be able to predict what the concentration of API would be in now and for the next few minutes. So you can see some examples on using the model to see whether the system can reject disturbances from feeders for a given compound or formulation. I think there's links to what Tom was saying earlier, that if you have a certain 10% fluctuation at the end, doesn't mean that you can only have 10% fluctuation at the beginning with your feeders, because of course these blenders and the feed frame provide some dampening of those high frequency disturbances from the feeders. And it was also used to demonstrate that you could use this model to predict not only the current concentration of API at this time going into the dye, but also the prediction of what was going to happen in the future. So this gives you, together with a physical, say, NIR probe, gives you the ability to look at not only what's happening now, but what is likely to happen in the future, which allows you to develop more robust control strategies that take into account expected future behavior. Okay, very quickly, something on the importance of model and data quality. We are, so I've taken this diagram from Tom's presentation, this verification validation 40 charter. You indeed, our approach is also very much based on, you need to define your context of use first. If you're going to use the model mainly for interpolation and you've generated quite a bit of data, you're probably, your parameters don't need to be as accurate or have small confidence intervals as when you're going to do extrapolations. So it's good, also a good thing about the goal of modeling. So one approach is to use the modeling mainly to determine the critical process parameters that you're going to use in a physical DOE. So physical DOE is essentially QBD 10. And you could say, well, I'm going to use the model to provide a more objective and a better reasoning to determine which of my 5 or 10 factors I'm going to use for a 2 or 3 factor DOE. So as long as you can prove that that approach is going to be a lot more robust than something that is quite subjective, based on people's own experience and qualitative reasoning, that's one thing. If, on the other hand, you want to use the model to directly determine a design space, then of course we need to ensure that the effects of the uncertainty in the model are smaller than the effects that we're really interested in, which is the effect of process variability, raw material variability, physiological variability, as well as, of course, the different values that our formulation and process parameters can have. And so building on this virtual DOE framework we presented earlier, it's now possible to perform a quantified credibility assessment of the model and identify key causes of model uncertainty. So this next diagram shows you this is a diagram I showed you before, and we've extended it with a third category of factors, which are the model parameters with correlated uncertainty. And this uncertainty doesn't only result from the fact that the model may not be perfect. We may have made some assumptions, or we just don't actually know the exact equations to be used. Even if the model is perfect, if it's a very simple system, the fact that the measurement and sampling introduces variance and bias will result in uncertainty in parameters. So actually this framework is not just about determining whether the model is good enough, it also says something about the quality of the data, something that we may not always understand if we use a purely data-driven approach. Okay, so that is a capability that is also now available. It hasn't been used in any case studies yet. We're obviously very keen to work with some leaders in the area to see how we can do it. But if the result of this virtual DOE, including the parameters and the uncertainty in these model parameters, if the outcome is that the largest sensitivity is to one or two of the parameters, we can then use that information to design additional experiments to reduce the uncertainty in those parameters. Okay, I need to wrap up. So concluding remarks. So over the last, I think, decade, a lot of progress has been made in developing mechanistic models for the various drug manufacturing unit operations and also product performance models. In particular in the small molecule area, I think the large molecule area is catching up. Certain areas in large molecule, good mechanistic models can be developed, but in many areas there's a need to go for more hybrid approaches where we try to use the data-driven aspect only for the parts we don't know, so that we need not too much data and more transferability. There are quite a lot of pharmaceutical companies using these models now, mainly in the product process R&D and tech transfer areas. And I think the key thing now, and this of course links to the first part of the talk, is that we really need to have good methodologies to make sure that we account for the quality of the model as well as the quality of the data used to calibrate the model in order to be able to use these models with known confidence for setting specifications and control strategies. And I think as one, as a modeler, one of the things that we typically miss from data is that most data sets don't have much information on the variance, the accuracy of that data. If you don't know that accuracy of the data, you don't really know how accurate the model is. Even if the model is perfect, if the data is not very accurate, the parameters are going to be less accurate. So we really do need to have that information. Okay. And with this enhancement to the virtual DOE, I think we can go from a qualitative credibility assessment to a quantitative credibility assessment. Again, trying to take out as much subjective elements as possible to ultimately ensure better pharmaceutical quality. Okay, thank you. And I'll hand over to Stephen, I think, for questions.
Thank you, Dr. Bermingham. So yes, now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question will be, what are the types of computational models the FDA has seen for use in specification setting and establishing control strategies. And that'll be for you, Dr. O'Connor.
Yeah, I think as I tried to indicate in the talk, we are seeing more of these hybrid, and I've talked a little bit more about the process models, which is kind of as the models come in to Sean's talk. But in addition to those, we're also seeing a lot in the performance area, actually very heavily, like compartmental type of PK, PD models used a lot to set, try to set more clinically relevant solution specs. But in the process side, where we're trying to talk about in-process controls, we've seen like reaction models, the kinetic models. Again, with the RTD models, we've seen a lot. We've also seen particle population balance models. So those are kind of applied to granulation and crystallization processes, but they've been helped to set some of the process parameters limits in those processes. So I think, yeah, we've seen kind of a wide range of model types, and we kind of continue to expect to see more as the capabilities and tools improve, as more companies start using them, and they just get more used to kind of including that as part of their justification in the regulatory filing.
Thank you. Our next question is, you mentioned the SBP Alliance on the vision slide. Can you tell me more about this alliance? I believe that's for you, Dr. Bermingham.
Okay. Thank you. Yes, actually, you mentioned the SBP Alliance briefly in my introduction, Stephen. So the SBP Alliance is a pre-competitive alliance that was founded by Lilly and Pfizer and PSE in 2013, and its goal is to accelerate development, adoption, and regulatory acceptance of the systems-based pharmaceutics tools, which is essentially this integrated view of drug manufacture and product performance. Since we started the alliance in 2013, we've had GSK and Roche join us. It was in December 2016, and one company that was not in the introduction because it's quite new is that Sanofi have also joined the alliance. It's an alliance that focuses in the first two phases up to about a year ago, mainly focused on the physics and chemistry to include in models, and now the focus is much more on the workflows of how to calibrate models, how to do model discrimination, how to do all these things robustly and efficiently. But the virtual DOE concept is something that came very much out of the SBP Alliance, and yeah, I'll leave it at that. If you have any more questions, of course, feel free to contact me.
Thank you. Our next question is for Dr. O'Connor. Will the expectation to include V&V evidence for all models in a regulatory file, including low-impact models? And that's you, Dr. O'Connor.
Yeah, so that would not be the expectation. We're still following the ICH points consider guidance, which does outlay what kind of information needs to be submitted as part of the filing. I think the standard could still be helpful internally for documenting the evidence you generate even for low-impact models. But as far as putting in the filing, no, that would not be the expectation. Generally, for low-impact models, we just describe the model and its role in decision-making and don't supply that kind of additional data.
Thank you. Moving on, our next question. We often see models for a small molecule drugs. Can you talk about applications for biological drugs, both manufacturing processes and formulations? And I believe, would Dr. Bermingham, would you like to answer that first?
Okay, yes. So as I mentioned, most of the work so far, at least from the PSE perspective, has been small molecule. If I just go through, I do have a few slides, or not a few, I only have one actually. But so we have started doing some work in the area. Where is it? About 2 years ago. So just 1 second. Here we go. Yeah, so PSE received a UK government grant to start developing some models for bioreactors and chromatography. So you can see on this slide that there is a prototype available. It's being tested by two industrial partners at the moment. So we are making some progress there. Obviously, still a lot more unit operations. We have also already developed capabilities to be able to link data-driven models and mechanistic models so that we can use the hybrid models, which I think everyone probably agrees is necessary, especially in this area. I think the good news from our perspective is that a lot of the platform functionality, so the virtual DOE capability that featured heavily in the presentation, how we do parameter estimation, internal, external model validation, and so on, model discrimination, that these are all sort of agnostic to whether it's small or large molecule. So our focus over the next few years will be to extend the number of models that we have for large molecule, and ultimately, again, it's going to be more about understanding what are the best workflows in large molecule to make use of models, combining mechanistic understanding and data-driven approaches.
Thank you for that answer. Our next question is, how can pharmaceutical manufacturers interested in using process models as part of their control strategy engage with the FDA? And that'll be for you, Thomas O'Connor.
Yeah, so the FDA and CEDAR particularly has what we call an emerging technology team. And then that team provides an avenue for sponsors looking to apply kind of novel technology and approaches. And we had several sponsors come to us to talk about their modeling approaches, especially when it gets into the realm of how they're going to control product quality. And we discuss a lot about these questions, about validation and just better understanding their intent. And it helps us to learn from them and provide some early feedback that they found very helpful. So the emerging technology team has a guidance, that's a process guidance that tells you how to send a request to the team and what kind of information we're looking for as part of that request to get a meeting with the FDA and discuss these questions. So that would be my recommendation, to engage the ETT team.
Thank you. Our next question is, did you implement a virtual mechanistic DOE as input for a machine learning algorithm yet? And the final goal could be an online control strategy independent of huge computational resources. Sean, should I give your thought first?
Sure. So we haven't done it yet, but we have actually, I think the next release of of our software will have the ability to indeed use the virtual DOE capability to create a very large data set from which you can build surrogate models. Things like the Alamo approach, I think, from Carnegie Mellon. So this is, so it's a very good question. It's actually exactly what we want to achieve. I think there are people actually who have used, especially in academia, who've used some of our tools, who have run these virtual DOEs and then done their own post-processing with the data sets to come up with surrogate models. And we think that's really important, especially for online applications and or applications where robustness, of course, is key. So again, if whoever asked the question, if you're interested in testing this, I was happy to work on some case studies together. We should have this surrogate modeling capability in our platform. I think it's released later in the year, but definitely still this year.
Thank you. And with that, unfortunately, we look like we may be running out of time for questions. But I would like to thank the audience members for sending your questions in. Before I finish the webinar, I would like to ask our presenters, Dr. O'Connor and Dr. Bermingham, if they have any closing remarks. Dr. O'Connor, would you like to go first?
So I would just say I'm chairing a meeting through AICG on advancing process control in the pharma industry. It's at the end of September 30th and October 1st, and lots of process control, process modeling and process understanding, really from the understanding of the basis of that. And at that meeting, you'll hear a lot, both large and small molecule. So I think if people are interested, they can look at that meeting.
And Dr. Bermingham?
Yes, I also have an event to plug. So we have actually in the US, in Tarrytown, New York, on 10th to 12th of September. There is an Advanced Process Modeling Forum, which is essentially a PSE user meeting. This will all be life sciences related. Tom will actually be giving a keynote there. There will also be presentations from, I believe, Biogen, GSK, Amgen, Pfizer, and probably a few other companies. I think this will be if you're interested in finding out more about these kinds of technologies and talking to practitioners, people from the pharma industry, then this could be a good opportunity. Also opportunities to get your hands dirty on the software and play around with it. I have just posted a link to the event, or at least I think I have, if I know how this works. So if you're interested, please have a look at the link or contact me to find out more.
Great, and I would like to take this opportunity to thank our presenters. Once again, that was Thomas O'Connor, scientist at the FDA, and Sean Bermingham, head of Formulated Products at PSE Limited, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand webinars. And if you are watching this on demand or had your questions sent in but we hadn't answered it yet, feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.